Understanding base hammer. Okay, before we start looking into this paper, there is some background knowledge that is presumed here, and I will not have time to go into this, but I just mention it. First of all, it mentions the k-means clustering algorithm, and this was actually a topic in BIN310. So if you like, you can go back and watch some video there where I talk about k-means clustering. This is in Norwegian. But you find also other, you can just search for it, right? Googling, you will find some explanation of this algorithm. Yeah, it's part of this method. We also talk about likelihood functions in this paper. And this is sort of an idea from statistics. Some of you may have heard about the likelihood functions and maximum likelihood estimation. This is sort of very basic statistics really, but I'm not sure the statistics courses at NMBU talk very much about it. And also this concept of overfitting is quite important I will not dig into it here, but it's mentioned in this paper. Those of you who have taken courses at data science, you will know what I'm talking about here. It's about fitting a too complex model to too few data. Right? That's the problem of overfitting. And there are many ways to avoid this. Cross-validation is the most used one, but there is also something called the Akaike's information criterion. And what is used in this paper is a variant of this called the Bayesian information criterion. So in case you find this in the paper and you don't understand what it is, well, you can, if you want to find out, Google the overfitting concept, and you may get some explanations. I will not have time to do it here now. Let's see how this base hammer procedure is outlined step by step. So what is it actually doing? It takes some reads, right? That's the raw input here, directly from the FASTQ files. And it computes some k-mer statistics. Actually, it just collects the k-mers, I would say. That's the first step. Then construct something called a Hamming graph and the connected components, and this we will come back to. This is a very important step. Then there is a Bayesian subclustering of the connected components, and here is where this k-means clustering comes into the picture. So subclustering is the important thing here. Then from this they collect something they call solid k-mers. So we will see what this is. And then from these solid k-mers, we can sort of expand the solidness in step number 5. We will also see this is pretty easy. And finally, correcting the reads, right? So the point is to take a set of reads as input, a FASTQ file or 2 FASTQ files if we have Illumina data. And the output is also FASTQ files. So we start out with some reads and we end up with some reads, but the point is that we now will try to eliminate sequencing errors in these reads. Also, the base hammer will discard some reads, but that's really not mentioned in this paper. It only talks about how reads are corrected, and it sounds like if you have like 1 million reads in, you will also get 1 million reads out. This is not the case really. Some reads will be discarded. They are impossible to correct, we could say. And that's really not mentioned, but that's when you run base hammer, you will see that. Well, let's dig into these steps now and see a little bit on how this is working. But before we do that, let me say a few words about notation, because you may not be used to reading this type of literature, right? This is scientific papers in bioinformatics, and they use sometimes quite extensive mathematical notation. For instance, alphabet. The capital sigma is the alphabet, which is actually the symbols that we work on here now. So this is, since this is reads, it's the DNA alphabet, right? But alphabet is something that you can find mentioned in many papers of this. And it's usually either the DNA or the RNA or the amino acid alphabets, right? But this is quite typical of this paper, that they talk about the alphabet as a set of symbols. Then, of course, k-mers. This is the really basic quantity or element in this paper. So a small x here symbolizes a k-mer now. So this is one k-mer collected from one particular read, right? X. And also the elements of this X is indexed from 0. And this, I think this is a little bit strange. But when you count elements in a vector, in R we count from 1, right? The first element is element number 1. Second is element number 2, and so on. But in many programming languages, the counting starts at 0. Python, for instance, is also counting from 0, I suppose. So they have chosen to count from, I think this is really silly, but okay, it doesn't really matter. So this is element number I in the k-mer, right? And they use the square brackets just like we do in R, actually. So this is the same way of expressing an element as we are used to in vectors in R. So this is element number I, and remember now I starts from 0. So if you have 10 elements, it's element number 0, 1, 2, 3, 4, 5, 6, 7, 8, and 9, right? Not 10, because it starts at 0. That's, I mean, no one counts like that. So why did I choose to do it? Okay, this is also very similar to R. You have an index vector I here with different positions. This could be position number 1, 3, 8, 6, 5, anything, right? And then you refer to those elements in the k-mer like this. This is a range, as we call it, right? So this is different from how we do it in R. In R, we would type I colon j from position I up to position j. But in this paper here, they write with a comma between, right? So this means from position I and up to position j, all the ones in between as well. And finally, Q is now the quality scores, right, for the k-mer X. Because remember, the input is from FASTQ files, so each sequence, each DNA sequence also has a corresponding quality sequence. So if you have a k-mer from a read, you also have a quality k-mer, we could say, from the read. So this just symbolizes this quality. But actually, it's not actually the quality scores, it's more like the probabilities, as we saw in the previous module, that we can always convert these quality scores to probabilities. So it's actually the probabilities that are used in this paper. And then they say for each unique k-mer, they keep sort of 3 pieces of information for each k-mer. The count, how many times did you see this k-mer? The quality, that's this one here, as an average of all the k-mers who are identical. And then the error vector, which is yeah, that's this one, yeah. The single elements, right, for each symbol in the. But I have to say this part here is not actually used, as far as I can understand. It's not really used at all in the description of the algorithm. So we look at single k-mers and not all the unique ones, because once you start looking at unique k-mers, in the case you have multiples, seeing the same k-mer over and over again, you need this count. but in the formulas we will see no count. We will only see the single k-mers. So the first step is simply to collect the k-mers from the reads, and this is in principle the same thing that we did with the jellyfish software in one of the examples in this module. So collecting k-mers, they describe this very sort of detailed in the paper, but it's more a technical way of how to collect the k-mers in a fast way. This is not so important, I think, to understand why this is super fast. Just realize that it's all about collecting k-mers, really. What I do talk about, and I would like to mention here, is hash map. They say that they use a hash map to do this. So what is a hash map actually? And this is, I suppose that some of you have heard, if you're taking some courses in algorithms and data structures. I know there is one at the data science, but most of you have never heard of hash maps, I suppose. What is it? Well, the idea of a hash map is that each canonical k-mer is converted to an integer by some function, right? So think of it as a function. You put in a k-mer and out comes a number. And this function must be designed such that each unique k-mer give you a unique number. But if you put in the same k-mer, you get the same number, right? But each unique one produces a unique number out. And why is this useful? This is used a lot in what is called hash tables. And they're used for very super fast lookup. Imagine you have collected k-mers and you collect yet another k-mer and you wonder, have I seen this k-mer before? If you have to search among the k-mers, it's quite a job to search among all these k-mers to see if you find the same as you now have collected, right? Even if you sort them alphabetically, it takes some lookups to find if you have seen this one before. If you have a hash function, on the other hand, it takes you one single lookup, because then you have, if the number between the k-mer is, let's say, 10,300, then you have made yourself a vector, and this k-mer is stored inside element number 10,300. So you can go directly to the cell in that vector where you have the hash number, and you can look inside and see, have I seen this k-mer before? Or even how many times have I seen it before? So it's a super fast lookup. The price to pay here is this hash function, right? Because it has to sort of map each unique k-mer to some number. It doesn't matter which number as long as they are unique. And they shouldn't be infinitely big, right? Because then your table here must be very big to have cells of that. Yeah, if the number gets many million, you need many million cells in your table. Yeah. So again, it's very sort of mathematical notation. How do I read this here, for instance? I read it like this. The hash map function h is such that it takes the space of all canonical k-mers. That's what this symbol means. You see the alphabet to the power of k. It means the space of k-mers, or the set of all possible k-mers, and maps it onto the positive integer, or non-zero, non-negative integers. This is capital Z, right? In mathematical notation. And it's even a subscript n indicating that no bigger than n. So these are all the integers from 0 up to n. So this is a very short and precise description, but it's a mathematical language. You need to understand how to read this very short and exact statements. Let's have a look at Hamming graph, because that was step number 2. So now we have collected all the k-mers. So what do we do to those k-mers? First of all, what is a Hamming distance? And here is again something I copied from the paper. And this is a very short and very exact description of what a Hamming distance is. But you need to understand the language of mathematics to read this, right? So what does it actually say? It says the distance between k-mer x and k-mer y equals, and what is here? First, let's start sort of in the middle of the expression. There are some curly braces here, right? And that always means a set of something. And here we have the set of I inside this range, right? From 0 up to k minus 1. So for all I inside this range from 0 up to k minus 1, we select those who are such that the element number I in the first k-mer is different from element number I in the second k-mer. So this here is actually a way of saying, look at all the positions in the k-mers where the k-mers differ. Not equal, right? Only those positions where these two k-mers differ. So these curly braces is this set of positions. And then you have these vertical bars on the outside, this is sort of the absolute value, which is cardinality of a set. So it's actually the number of positions. That's what it means. So a Hamming distance between two k-mers is simply how many positions do they differ in. Count them. So the Hamming distance is always an integer. If they are identical to each other, the distance is 0. There is no positions with the difference, right? If there is difference in one position, then the distance is 1. And so on. 2, 3, 4. You can have up to k differences, right? Since these are k-mers. If all positions are different, then the Hamming distance is k. So we can in principle compute this distance between all pairs of k-mers. And of course, that's a horrible job. So that's why in the paper they describe in detail how they do this without actually doing that pairwise comparison between all. Remember, we may have millions of these k-mers, right? But that's not important. The technical part that describes how this is done quickly is not so important. It's first we must understand what they are doing. Then we can be concerned about doing it quickly. Okay, they define a distance threshold, use this Greek letter tau, and say that we make now a Hamming graph. And what is a Hamming graph mean? And here is figure number 3 from the paper that I copied with some pictures or images of two Hamming graphs here. This is Hamming graph with distance threshold 1. So here tau equals 1, and over here it's 2. But let's look at this one first. So here we have a number of reads, right? The first read is this one, and from this one we can collect some k-mers, and here k is 5, right? So we have 5 mer. These are very short reads, but this is of course just examples. So from this read here we collect these 3 k-mers, and we put them into the graph. And the same here, we have another read, collect some k-mers, and the third read, collect some k-mers. And then we can see the k-mers here and down here. And then you can see some of them have been connected by edges, that we call them. Right? This is a graph way of displaying things. A node, each k-mer is a node, as we call it. And between nodes there can be edges. And why are there edges between some of them? Those who are connected with edges now are those who differ with Hamming distance 1. Right? So between this k-mer and this k-mer, there is distance 1. There is one element, one position that is different. And it's the third position I can see. There's an A here, but it's a C here. And apart from that, they are identical. And again, another edge here because there is one difference from this one to this one as well. And again, it's a third position. Goes from A up here to a G down here. So again, there's only one difference and an edge is drawn. And that means we also have an edge here because again, it's the third position that these two disagree with. And then you have an edge down here, but now it's a different position, right? Because now it's a second position. And this one is not, this is more than distance 1 away from this one because these two have two differences between them. So there is no edge here. Because now we set the threshold at 1, right? That's why you get one part of the graph is here. This is what we call a connected component. So connected components, that's the part of the graph that is connected. You can sort of walk from one k-mer along edges to another k-mer. They are connected. These three are also connected. And this one here is a singleton, we call it. It's alone. It's too different from all the others. And here you get a similar thing, but here the threshold is 2. So now there will be more edges, right? That's the only difference really between. So the more, the bigger you make the threshold, the more all k-mers start to connect with each other, of course. But let's stick to this threshold 1. And why is this Hamming graph actually a good idea to start thinking in terms of? And that's something that was not really communicated, I think, in this paper. So let me try to do that. Assume now we have in our data set sequenced with a a k-mer coverage of 50. Right? We have seen the concept of k-mer coverages. And what does this mean? It means that if k-mers are without errors, we expect to see them around 50 times. That's what this means. If the k-mers, if there is no sequencing errors in our reads, and we collect the k-mers, each k-mer should be around 50 times observed. So let's say this is one of those error-free k-mers. And I made it red here to symbolize that this one we have seen many times, right? There are actually, you can think of many k-mers sort of on top of each other here. But since we also have sequencing errors, we will typically have some variance of this k-mer, which is due to errors, right? So even if this is sort of the normal error-free version, there will be from time to time in some reads this variant of that k-mer, or this variant, or this variant of the same k-mer. Or if all the errors occur in the same position, it will look like this, right? Where we have this. And again, I connect now everything which is difference in 1 and no more. So this is the idea of the Hamming graph, that you will in a connected component like this typically have one k-mer which is sort of the true one, the error-free one, and around it as neighbors you will have some variants due to sequencing error. But these will be connected, right? Because they are not that different from the original. And in this case, we draw the limit at 1, but you could draw it at 2 or 3. This depends on how long the k-mers are, right? But remember this, that there is sort of one central k-mer which is very frequently occurring, and the others are rarely occurring. Think of it as a mother sequence, and then all the other ones around are sort of generated or children of this mother sequence. So that's why I draw this red to sort of make it stand out from the rest. So what we are trying to do now is to, by making these Hamming graphs, try to locate these k-mers among all the k-mers, right? Then they start in step 3 doing clustering. And now we look at each connected component in the graph and try to split it into smaller pieces. So first of all, consider this connected component, the consensus k-mer. What is this? If we go, let me go back to the previous slide actually, to see if we can understand this consensus k-mer. Imagine now we have 50 copies of the red one, and we have 1 copy of each of those black ones. The consensus sequence for this connected component here is the k-mer that has the most votes. If you go position by position and vote among the k-mers which symbol should be there. Of course, in the first position there should be an A, because this one gives 50 votes to A, and also this and this and this votes for A. So there will be 53 votes for A and only 1 for G, 1 for C, and 1 for T. So the majority says A in the first position, right? There will also be a C in the second position and a G in the third position. And this is because this mother sequence has 50 copies, right? So this will have the majority in all positions, regardless of how the other ones look like. But of course, in some cases, the center here doesn't have 50 votes. It could be it has only 2 or 3, right? Because even if we have a k-mer coverage of 50, we know that there are some rare cases where coverage is much, much lower. So anyway, regardless of how it looks like, we compute the consensus sequence, the majority vote for the connected component, and then we consider all the k-mers, all the other k-mers, you could say, or all k-mers really, both those in the center or anywhere, wherever they are. Then we define a likelihood function for this. And what is a likelihood function? The likelihood function says something about a probability. It's the probability of seeing these k-mers given that we have the consensus sequence, right? And that's found by simply multiplying together these error probabilities. So if there is a difference between the k-mer and the consensus sequence, we presume it's due to sequencing errors, so we multiply the probability of sequencing error in those cases. If, on the other hand, the k-mer is identical to the consensus sequence, then there has been no sequencing error, so we multiply 1 minus the error probability instead. This is the probability of not having an error, right? And these capital pi's here, they mean product. So if you're used to summation symbols, the sum, which is a big sigma, this is a product instead. So it actually means we multiply together these probabilities, either probability of error or probability of not error, depending on if position number J is different from the consensus or not. So it's simply a looping over all the positions, multiplying together, and then you get the likelihood. And notice now, if this k-mer is equal to the consensus sequence, and the quality scores are very good, so this probability here is very, very small, so it's essentially 1 here. Then you multiply together 1 times 1 times 1 times 1, which is 1. Right? But as soon as you come here, if there's a difference between the k-mer and the consensus, then you multiply everything with this one, which is also usually a quite small value. Even if the quality score is rotten, this probability is not super small, right? So this likelihood function will be bigger for those who look similar to the consensus sequence and smaller for those who are different. And now we start splitting this connected component into subgroups. And actually the way we do the splitting, That's the k-means clustering, but that's really not important. Think of it, this as just some random splitting into subsets. So we have m subsets, right, in this case. And then in each of these subclusters or subsets, we again find the consensus sequence, right, in the same way as before. And now the likelihood for these subclusters looks like this. So the likelihood for this subclustering is simply the likelihood from up here for each subcluster, each member of each subcluster. We compute the likelihood, right? And then we multiply together these likelihoods for all the members in that subcluster, and then we multiply over all the subclusters again. So it's actually more multiplication of probabilities, this. So why do we actually do this subclustering? And that's where I need another drawing. So we saw previously that we can have typically something like this, right? Where we have one k-mer that occurs many times. This is the sort of error-free version. And then you have this with errors spreading around it like this. But of course, there could also be another mother sequence that looks like this. Another k-mer with 50 copies, right? Occurring very often. And then we can see that this particular one here, this child here, could be generated by one sequencing error from this one, but also with one sequencing error from this one. It's impossible to know which group this one belongs to, right? And we can see that since we now have one error here and one error here, it makes all of them connected, because we now have this edge here that connects both this into one connected component. And we can easily have many red sequences connected like this, right? And obviously this is the picture we want to try to alter now, because if we now compute the likelihood for this as it is, it means that we have to now take, first of all, we have to find a consensus sequence for this one. And what is that? Well, in the first position, it's an A, because these two mothers agree that it's an A, and they have the most votes, right? But in the second position, this mother says C, and this one says G. So now these children, and if this has 50 votes and this has 50 votes, now it depends on the children. And in this case, it turns out C, right? Because there are more children over here having a C in the second position than there are over here having a G in the second. So it's pretty random whether it's a C or a G. But anyway, the likelihood for this k-mer here now becomes super bad because it has a G in the second position. And the consensus sequence is C. So if you go back to the previous slide, you will see that this one has now a mismatch to the consensus, right? And since it occurs 50 times, it will be multiplied 50 times, right? So the likelihood for this one in particular, and also for these three, will be super bad, and the total likelihood for the whole cluster is also rather rotten because we multiply this over all members, right? If we now split this into two groups, like I indicate with these rectangles here, and this looks like a very smart way of splitting it, right? Then we will have another likelihood that looks like this. So now we have two groups, C1 and C2. In each group we find a consensus sequence, and that will now become the mother sequences because they now fall into each group, right? So this is the likelihood for the blue group here now, and this is for the brown, right? And none of the likelihoods are very small now, because now every member here looks very much like the consensus, and over here also every member here look very much like the consensus. So now both these become big, and this likelihood becomes much bigger than the previous one. So this turned out then to be a very good clustering, because this likelihood, it measures sort of the goodness of the clustering. The larger the likelihood, the better the clustering. So this is the idea of the clustering step here now, that try different splitting of the data, compute the likelihood, and see if it increases. And if you can find a bigger likelihood, you have found a better clustering. Right? So it's a matter of just searching for a better split of the data. And you can think of it as a random search, but it's not really random. That's where this k-means clustering algorithm comes into the picture, but we will not dig into that. But just understand this idea here, that it's a good idea to actually split here, because now we have a cluster here that identifies this mother, and we have another cluster here identifying this one. And if we had them merged together, we couldn't find more than this one, right? Solid k-mers. Yeah. So let's say we have found the optimal subclustering. And then we compute for each cluster the quality. And this is a simple formula here. First, you have to compute this one, which is just a matter of multiplying together this the probabilities of error. And notice we take 1 minus, so this is actually the probability of not having an error. So for each k-mer, this is the probability of not having an error. And then this is used inside this formula here. So for each k-mer in the cluster, we take 1 minus the probability of not having an error, which is the probability of error, and then 1 minus this again. So again, this is sort of the probability for the whole cluster of not having an error. Right? So if this one is big, then this one is also big. And this is the quality of the cluster that is in this algorithm here, in this Bayes hammer method. And they say that if this PC is above some threshold, then we call the consensus k-mer of this cluster a solid k-mer. That's something we can trust, right? And these are typically the mother sequences that we saw previously, right? The consensus k-mer is a solid one if these error probabilities are small enough, you could say. So remember, this comes from the FASTQ file, right? So if you have a high quality read, and many of the k-mers come from high quality reads, then you have a high quality cluster. And then they expand this solidness in a really simple way. So imagine we have a read here. We have all the k-mers from that read. And then after this clustering and blah, 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 we find that some of these reads are solid, right? Marked in red here. And notice now that this red, they cover the whole read. Because there's one here at the start of the read. There's one in the middle, which is overlapping with the previous red one. And then there are 2 red ones at the end here, overlapping with the one in the middle. So all position in this read is covered by some red, right? And then they say, okay, then we call all the k-mers in that read solid. So even if only the sort of original cluster consensus sequences are solid, in this way, because solid k-mers occur on the same read, we say also that the other are solid if everything has been overlapped here. So this means we have a large set of solid k-mers in the end, and this is the last figure. This illustrates now how we use this for error correction. So here we have a read, and here are all the k-mers from that read. Right? Now here we have the gray ones here are the non-solid k-mers. So and here you can see 2 of the k-mers here are actually members of a cluster who has a solid consensus, right? And then we actually just replace the original k-mer here with the mother, you could say, in the same cluster where this one belongs, right? And also here. And also here. And then we can see by now making majority votes in each position, this T here that was originally in the read is now being changed to an A down here. So this is how the read looks like after base hammer has done its work. Because in this position now, there are 3 votes for A and only 2 votes for T. So this is how we could correct this error. This is sequencing error. At least that's the conclusion of base hammer. It must be a sequencing error because we see that the solid K-mers, they tend to have A's here in that position. What I did not really understand in this figure, and what you can also discuss if you like, what are these black K-mers here? They are seemingly solid. And then and they contain this T, right? Here, this error T, right? How on earth could these be solid? Because they should also belong to some cluster, right? Why is the cluster that these belongs to, why doesn't they have a solid mother sequence that has an A here? Well, there are some possibilities, but at least I would say that for pedagogical reasons, this was not a very good choice of example for illustrating the error correction idea, I think. But that's something we can discuss if you like. How is it possible that these 2 K-mers are solid without having corrected the T here? Because, I mean, since these solid K-mers appear, there must be a number of other reads covering this region, right? Giving a rise to these K-mers here. They must come from some other reads, right? That covers the same area here. So why don't we see the same here? When you have red solid up here and also one down here, how can these 2 in between not be also red? Yeah, okay. But now we have gone through the whole procedure really and see how we can start out with a read and do the error correction, come out with a different read due to this Hamming graph clustering.